Welcome to this presentation on the importance of codons and anticodons, the tiny but mighty elements that translate the language of genes into functional proteins. This is your cell and molecular biology course, and I'm your course instructor in Center. Over the next few minutes, we will unpack how codons and anticodons work and why they are crucial and their role in keeping life going on at the molecular level. So we will be seeing all these in the next few minutes. At the heart of life lies the genetic code. The DNA stores instructions, but it's the codons, that is the three-letter sequence in the messenger RNA or mRNA that carries these instructions to the protein building machinery. Each codon specifies an amino acid, the building blocks of proteins. This code is nearly universal, that is, it is shared by almost all living organisms, which makes it fundamental to life itself. So, what are codons? Codons are sequences of three nucleotides in the mRNA, and how mRNA is synthesized we have seen in the transcription video. That is, the mRNA is transcribed from the DNA. So, in case of codons, for example, the codon AUG codes for the amino acid methionine and also serves as the start signal for protein synthesis. We have seen that in the translation video. So there are 64 possible codons formed from the four RNA bases, that is adenine, guanine, uracil, and cytosine. Among these, 61 codes for amino acids, while three serve as stop signals that mark the end of a protein chain. These stop codons include UAA, UAG, and UGA. So this is about codons. Then what are anticodons? Anticodons are found on molecules called transfer RNA or tRNA. They are complementary to the codons on the mRNA strand. This pairing is crucial because it ensures that the correct amino acid is brought into place during the process of protein synthesis, that is, translation. Without anticodons matching the codons, the entire process would be chaotic and inaccurate. So we have seen about this in the translation video. That is how, after leaving the nucleus, how the mRNA is brought into the protein synthesis unit. That is, which includes ribosomes, that is smaller and larger unit of ribosomes, and then the mRNA. And then we know that tRNA brings the specific amino acid based on the codon present in the mRNA. And this tRNA has three nucleotide, which is the anticodon, which is nothing but it is complementary to the mRNA codons. So it will bring a specific amino acid to the site so that so that an accurate protein which is coded by the gene is produced. So this is how it works. So now let's see how codon and anticodon pairing works. The pairing between codons and anticodons follows specific base pairing rules. That is, adenine pairs with uracil and guanine pairs with cytosine. Interestingly, the third position of the codon allows some flexibility known as the wobble position. You can see in the illustration the third position of the codon, which is the wobble pairing. So this flexibility helps cell make proteins efficiently even with a few tRNA types. According to this concept, the third position of the codon allows some flexibility or wobble in the base pairing. This means that a single tRNA anticodon, that is, a single tRNA anticodon, can recognize and pair with more than one codon which is present in the mRNA. This flexibility is essential because it lets cells use fewer tRNA types while still reading that is all the 61 codons that specify amino acids. The wobble ensures efficiency and speed in protein synthesis without compromising accuracy. So this is the wobble hypothesis. This is how the codon and anticodon pairing occurs, and in some cases, the third position is flexible so that more uh, efficiently the proteins can be produced. Now let's see the start and stop signals. The start codon AUG signals the ribosome to begin translating the mRNA into a protein. We have seen that the start codon is AUG. It not only starts the process but also codes for methionine, the first amino acid in most proteins. In almost all living organisms, this is the initiator codon, that is the start codon, which begins the translation process. And then the stop codons, which are, we have seen UAA, UAG, and UGA, or the stop codons, which signal the end of translation. This ensures that the protein is made to the correct length and it is not extended further. So all these is governed by the stop codons. So these are about the start and stop codons. Now let's see the role of these codons and anticodons in protein synthesis. During protein synthesis, codons acts as the instruction manual. The ribosomes reads each mRNA codon and the matching tRNA with the correct anticodon it will deliver the right amino acid. So these amino acids link together to form a chain, which then folds into a functional protein. So without this precise matching, proteins won't be able to form correctly. So all these, that is the ribosomes, codons, and the tRNA with the anticodons, and then it should bring the right amino acid. So all of these contribute to the production of a functional protein, which governs the functions of the cell. So what is the importance of accuracy? Accuracy in codon-anticodon pairing is very critical. If a tRNA brings the wrong amino acid, the resulting protein, which may not function properly, or it may worsen the condition. In conditions like that, it can cause disease. So cells have evolved proofreading mechanisms to minimize these errors, but even small mistakes can lead to significant problems. Thus, it highlights the importance of precise system. So accuracy is very, very important in case of protein synthesis and in maintaining the cell's integrity. The significance of codons and anticodons extends into medicine. For instance, a single codon mutation in the gene for hemoglobin causes sickle cell anemia, a serious blood disorder. So, understanding codon usage has also led to advances in gene therapy and also genetic engineering, and it offers hope for treating many genetic conditions. So, in the illustration, you can see the normal and sickle cell anemia conditions. In this, you can see the difference is in the nucleotide of the DNA. So when this nucleotide template is transcribed into mRNA, which is nothing but it is the complementary nucleotide of the DNA template, and then the tRNA, which has the anticodon and brings the accurate amino acid, will bring that amino acid according to the codon present in the mRNA. So with that, the from the normal amino acid which is to be produced, the mutant will produce valine. So this is how the codon mutation leads to genetic diseases. So codons and anticodons are the essential translators of the genetic code. They ensure that the information stored in our DNA is accurately turned into proteins, which are the building blocks of life and the functional units of cells. Or you can simply say the workhorses of the cell. So their precise interaction is the foundation of life and the key to future breakthroughs in medicine and biotechnology. So, not to forget the genetic medicine, which can find that is the knowledge about codons and anticodons can find cures for various genetic disorders. So, these are all the importance of codons and anticodons. Thank you for watching. See you in the next video.